Prepare to experience the strongest radio allowable by law. Secrets will be revealed, myths dispelled. From the studio gym where excuses never apply, it's Superhuman Radio with your host, Carl Lenore.
Welcome back to another episode of Superhuman Radio. We have an important show for you today. They're all important. When you really think about it, we really do produce some amazing content, way, way, way far ahead of anybody else. Today's going to be one of those shows. As you can see, the saga of my eye continues. I may end up having to go get a corneal transplant after all. After all that mess I went through last year trying to keep this eye functioning, the end result may be I just put off getting a corneal transplant, which I don't want because I don't want to have to use steroid drops in the eye. Then I'll develop a cataract, and it's just a slippery slope. So I'm going today and hopefully getting some good news. But before we do anything further, we have to thank our title sponsor, Legendary Foods. The coupon code is SHR10. The website is eatlegendary.com. They have the most amazing snacks in the world, and they have little or no sugar in them. Their tasty pastry is basically a Pop-Tart with 9 grams of high leucine protein and less than 1 gram of sugar. Top that. If you like Pop-Tarts or your kids like Pop-Tarts, that's the place to go. But check out their nut butters and their seasoned almonds. Stuff is amazing. EatLegendary.com, code SHR, 10% off. Show them some love. Well, now let me bring my guest on. Just give me 1 second. And we have to put up so my guest today is Dr. Eric Skaar. How you doing, Dr. Skaar?
Oh, good, Carl. How are you doing today?
Yeah, good. And so C. diff. How do you? I use the abbreviated name because it's easier for me to sound intelligent. But the actual word is what?
Well, it's an interesting question itself because it just changed. It used to be Clostridium difficile.
Right.
But it turns out this thing doesn't look exactly like the other Clostridium, so they changed it to Clostridioides difficile. But to make it easy on everybody, they kept the C for the first word in the name. So it's still C. diff. C. diff is the best way to abbreviate this thing.
So in the lexicon, when you put oid or oides after something, that means that it is somehow a precursor to or a pro to. Is that true?
That's the original intent is that, but sometimes the names get a little bit garbled. It's a pretty complicated process by which these things are named, as you can imagine.
Yeah, I know. And another reason why doctors don't have to explain things because patients can't read them anyway. So C. diff is a big problem in the United States, especially within the elderly that are institutionalized or hospitalized for long periods of time. Correct?
Yeah, that's right. This is the number one cause of infection now in hospitals. So the number one hospital-acquired infection causes a huge amount of disease in the United States and worldwide, and really bad disease too. So very high morbidity, very high mortality. It's really a nasty germ.
And the way the mortality comes about is that people get chronic diarrhea, they become severely dehydrated, and then that state, especially because we're dealing with a frail population typically, that pretty much pushes them into some other, it could be heart related because of electrolytes, it could be organ failure, but that pretty much kills them then, right?
Yeah, that's pretty much right. It's the massive damage to the gastrointestinal tract has profound effects throughout the entire body. And then often you'll get leakage from the gut into the internal organs. So bacteria that normally are contained within our gut can then leak and disseminate to other parts of the body. And so you can end up with gut bacteria in organs outside of the gut. And that's another way by which this can cause a huge amount of problems.
So this could actually, I mean, we could call this the end stage of this, a very aggressive leaky gut problem. Because we talk about leaky gut today, that people have different degrees of breaching of the lining of the gut because of a variety of different disorders. This is like whole scale, boom, the gut keeps nothing in, everything gets out into the bloodstream.
Yeah, you're looking at massive gut inflammation, and that leads to massive tissue damage, and then all bets are off as far as the barriers that typically contain some of these things. That's right.
Is the reason that we see massive tissue damage throughout the body because, 90% of the immune system is the gut, and the number one army of the immune system is inflammation. And so when the immune system becomes disrupted to that degree, the inflammatory process just goes crazy and starts killing. It becomes friendly fire. It starts killing everything around it.
This is a lot of it. So the pathogenesis or the disease process starts. At first, there's this whole process by which how do you get it in your gut? Why does it even take hold? And we can talk about that a little later. But the inflammation starts largely through the production of some toxins. C. diff makes some very famous, very nasty toxins. And those toxins cause considerable tissue damage. That tissue damage then calls in the army, as so to speak, the inflammation. And then once the immune cells come in to try to tackle the bacterium, they have to deal with the host has to deal with all these other nasty products that the immune cells make. So yeah, bystander damage is a huge part of this, just massive inflammation in the gut.
This I'm going to hit this again for a reason. The reason people die from COVID-19 isn't because of the virus. It's because the virus elicits such an overwhelming response from the immune system that inflammation attacks all tissue in the body, destroying the lungs. When we see high inflammation, we see thickening of the blood, increasing of D-dimer and thrombotic index. We see that in COVID-19. A lot of diseases today, the more of these interviews I do, I realize it's not the intrinsic effect of the disease, but the extrinsic effect of firing up the whole immune system and letting it go haywire on the body.
This is true of a lot of infections. So you always have overt damage directly, like we just mentioned with C. diff. There's these toxins, and the toxins directly damage host cells. That absolutely happens. But we have this super powerful weapon in the immune system that we mobilize against these organisms. But once you unleash that thing, it's hard to contain it, and you end up with lots of other problems, like you just mentioned. Absolutely.
So talk about your study. Why this study? What preceded this study that made you start to look at blood specifically as a enabler, if you will, or a cloaking mechanism of the C. diff bacterium?
Yeah. So my research program has always been interested in how bacterial pathogens get food when they're in our bodies. So how do germs that make us sick eat when they're inside us? And the food that we focus on are what we call trace elements or trace metals. So these are the same things in your average multivitamin: manganese, zinc, iron, copper, etc. So bacteria have to get these things in order to replicate, cause disease. And so that's a big part of my research program: is how do bacteria eat these foods, and how does our body try to keep them away, and how does diet impact these processes? So iron is one of the key nutrients that all these bacteria need. And it turns out the most abundant iron source in our body is this molecule called heme. So heme is what makes blood red. It's a molecule bound to hemoglobin, which is a protein in our blood to move oxygen around our body. But the oxygen is bound to an iron atom that's coordinated in heme. So all the oxygen, the ability of oxygen to move around our body requires this heme molecule. But because of that, we have a ton of heme, and because of that, we have a ton of iron. So if you're a bacterium and you want to get iron, heme is the best place to go. So we have an entire research program focused on how pathogens like staph infections, how these organisms get iron from heme in order to replicate and cause disease. And that's how we became interested in the molecule. And the Clostridium story is interesting because Clostridium doesn't seem to eat heme for use as an iron source. And that's where we got intrigued, because as we just talked about, this bacterium makes these toxins, causes massive amount of tissue damage, inflammation pours in, and a big part of that inflammation is red blood cells, which have heme carrying oxygen. So we know that C. diff had to be exposed to massive amounts of blood, massive amounts of heme during infection, but we also knew that it didn't use heme as an iron source. And so that led us to ask the question of, how does C. diff deal with encountering this molecule heme during infection if it's not using it for iron?
Let's stay with iron just for a second, because remember earlier you asked me what fascinates me? Well, for the past decade, I've been looking at iron overload, its linkage to senescent cells, and other things. I've done shows just about iron. Iron is rust. It gets in your body. Chances are you and I have the iron from a hamburger we ate as children still in our body. We don't have a system to get rid of iron. Now, there's lots of ways to get rid of iron, but it's not an innate system that's working all the time, like insulin manages blood sugar and stuff. And so we know that iron is highly oxidative. It's probably the most oxidative component or molecule in our body. In fact, a recent study has now linked ferritin not to iron content specifically, but an inflammatory response. It was just done in diabetic obese people. And so we now know that people with elevated ferritin may not actually have high iron. You have to look at TIBC and some other components to identify the actual content of the iron in their body. But could it be that the iron that is being liberated but not used is what actually goes out and damages all this tissue?
Yeah, this is, and just to follow up on your point, which is absolutely correct, iron is highly reactive. So this is what makes it so valuable, is it's a highly reactive element, which allows it to catalyze a number of really important biochemical reactions. But because
I don't know if you could, we dropped you, you lost the connection a little bit. Said at catalyze. You said iron is a highly reactive that allows it to catalyze.
It catalyzes a lot of enzymatic reactions. Its reactivity is what makes it so valuable, but its reactivity is also what makes it so toxic, as you mentioned. And it's even more toxic when it's a component of heme. So the nature of the molecular architecture of heme with iron inside it becomes extraordinarily reactive and extraordinarily toxic. And so absolutely free heme liberated at a site of infection, or anywhere for that matter, has the potential to cause a lot of tissue damage. And it also has the potential to kill bacteria. And so bacteria have to deal with this reactivity because they're running into, if you're a bacterium that causes inflammation, you're getting massive amounts of blood that pour into the site, red blood cells, hemoglobin releases the heme, and now you're surrounded by this highly reactive molecule that you have to deal with. And that's sort of the basis for this work that we're talking about today.
Okay, so you discovered that the C. diff is not using the heme as you would expect it to, or the iron, so to speak, but it's actually using it more to create a cloaking mechanism, right?
That's right. So the way that we did this is we just did a really simple experiment for us, which is to add heme to C. diff and then look at all the genes in the bacterium that change expression to say, what is this organism turning on or turning off when it's encountering heme? And we found a couple of genes that encode proteins that had never been studied before that the organism massively upregulates when it's encountering heme. And that we obviously became very interested in these proteins to try to ask the question of what might they be doing and why are they expressed at such a high level. And what we ended up finding was the C. diff bacterium, when it sees heme, it produces a massive amount of this single protein in the outside of the bacterial, the envelope is what we call it. That protein we named HSMA. For heme sensing membrane protein A. But HSMA makes basically a shell of heme. It's a heme protein complex around the outside of the bacterium. So this bacterium does two things for the organism. The first is it's now bound up all the heme. So it's no longer suffering the reactivity of the molecule, and it's not dying just from the inherent reactivity of heme. But then what it does is it uses the reactivity of heme to detoxify other oxidants that the organism encounters when it's in our gut.
So, this begs the question about if people are suffering from C. diff, it sounds like it's not a good idea to take antioxidants, or do I have it the other way around?
Well, it's so the way that we're thinking about this right now, and admittedly we still have a lot to learn, is when C. diff colonizes, it causes inflammation that brings oxygen into the gut. C. diff is an anaerobe, so it normally will be killed by oxygen. And one of the things that this molecule allows it to do is this HSMA heme complex, the shell. One of the things that it allows it to do is tolerate the presence of oxygen when the organism begins to see oxygen in the inflamed gut. Because since the organism's anaerobic, it's sensitive to oxygen. You would imagine if you had oxygen pour in through inflammation, well, all the bacteria, all the C. diff would die. But this protein prevents that from happening. So it's basically protecting it from the toxicity of the environment that the organism has created due to all this tissue damage.
Interesting. So when you discovered that the heme was being used this way by the C. diff, did you experiment with ways of trying to derail this process?
Yeah, this is something that we're doing now. So in a test tube or even in an animal, we know that if HSMA is bound to heme, so if C. diff has this protective shell, it's much more resistant to just the simple presence of oxygen. It's more resistant to reactive oxygen species that are produced by our immune cells to normally kill bacteria. And very importantly, it's much more resistant to antibiotics. And the reason for that, we think, is because one of the ways that antibiotics kill bacteria is they generate reactive oxygen species in the bacteria. So when a bacteria is getting killed by an antibiotic, it's stressed out and it starts to produce reactive oxygen species endogenously. And we think that HSMA protects the organism from antibiotics by preventing these redox active molecules from killing the bacteria. And so that has really important clinical ramifications because the amount of resistance that we see to like vancomycin and metronidazole, these are clinically relevant antibiotics, is really impressive. And we think if we could somehow sequester or capture heme in the gut during a C. diff infection, we'll make the organism much more sensitive to both the immune system and the antibiotics.
Is that how it enters our body, through the gut?
Yeah, so C. diff, the ability of C. diff to colonize is really cool and very different than most pathogens. So normally, if you have a healthy gut and you have a healthy gut microbiome, you never have to worry about C. diff. But if your gut microbiome is disrupted, usually with antibiotics, so what ends up happening is you reduce the diversity of the bacteria in your gut. So the number of species in your ecosystem is reduced. And now C. diff can come in and colonize. And it comes in as a spore, which is a very resistant type of bacterium. It's also very, very rare. There's only a couple bacterial pathogens that can sporulate into these highly environmentally resistant forms. Anthrax does this, C. diff does this. So it goes into your gut. It turns into what we call vegetative cells. So this is a live replicating bacterial cell, and it begins to replicate. But if you have a healthy gut microbiome, you don't have to worry about this. And so this is why the fecal transplant is such an effective treatment for C. diff infections, is you are replacing the gut microbiome with a healthy gut microbiome and pushing C. diff out.
So do we have have any samples been taken of people who seem to have reoccurring C. diff infections, and people who have had C. diff and it's never come back, and people who've never had C. diff, and try to isolate which of these microbes in our gut actually provide some sort of protective mechanism? So, I mean, I know my mother had fecal transplant in the nursing home because she had C. diff, and my niece lived in Arizona at the time. She was a family member, and she was on death's door, and boom, like 3 days later, she was better.
Yeah.
Do we know which microbes seem to be protective?
It's an awesome question. The answer is not really. So there's been some published work has come out that has implicated a few specific organisms as being more important to prevent the presence of C. diff or the colonization of C. diff or the virulence of the organism. But we're not yet to the point where we can create a cocktail, for instance, of only a few bacteria, and then instead of giving a fecal transplant, just give maybe a probiotic cocktail and prevent colonization. But I can tell you that this is big business. There are a lot of companies and academic investigators that are trying to do exactly what you just said, which is to create the minimal microbiome, so to speak, that a person can take to push C. diff out if they're sick.
So I have a couple questions that popped up. Obviously, the first one comes back to iron. What about reducing iron load in people who are prone to C. diff through a planned phlebotomy, for instance? And also looking at things like TIBC, not ferritin, but looking at total iron binding capacity of the heme and saying, okay, we want to get them down to a point where maybe the iron is contributing to some of the damage.
Yeah, this is a tough problem because the concept, I think, makes a lot of sense, right? If bacteria need iron, for instance, and you can reduce the amount of iron, you should be able to protect against infection. The issue is that metals are so important for how all cells function that if you are blindly supplementing or chelating metal from a person, you're not only affecting how the bacteria in their body are going to live, replicate, and cause disease, you're affecting how their cells and their immune cell is going to function. So I think that the concept of saying if we could measure the metal abundance of an individual person, and maybe they're anemic or maybe they have metal excess, and then do something about that to get them more to where we would expect a healthy, physiologically sound sort of level to be, I think that makes sense. I don't think it makes sense to just start blindly chelating or supplementing.
No, yeah, no. Yeah. And I'm not for chelation, by the way.
Right.
I'm for phlebotomy. Take the iron along with other components, because the blood has certain ratios that it likes. And if it doesn't get those ratios, it will steal them from bone or steal them from tissue. Or if it has too much of something, it'll stick it in tissue. And I think this is where the real problem comes. Iron accumulates, the body tries to manage it. Ferritin is designed to shuttle it. Ferritin can be elevated by taking vitamin C without the presence of additional iron consumption. So ferritin is a tricky one. We look at ferritin and people go, oh, no, your iron is fine, your ferritin's fine. But that's really not a good way to look at it. But I think that for people who are at that upper range of iron, knowing that when you were young, you were at the lower range of iron and you were fine, and I understand we're a bigger organism now, but just gently, once every 3 months, let's take a pint of blood. Let's see what happens. Let's take a pint of blood. And to the point where, because I do plan phlebotomy because I'm on testosterone therapy and I produce a lot of red blood cells as a result. And so, and I eat a pound of red meat every day and have done for 20 years. And so I do accumulate iron, but I try to keep it controlled. I don't want, because what happens is when the blood has a lot of iron, it starts sticking it in tissue. That's when everything goes wonky in the body. So I think planned phlebotomy could be an adjunct therapy for some of these people to see if they just don't have the reoccurrences once you get their iron levels just a tad down from where they are.
Yeah, I think iron excess is a huge problem. You're absolutely right. And managing, what you really want is you want to be at normal or the right level, so to speak, whatever the right level is, that allows your cells to operate properly while also not giving access to these bacteria. But we had a paper come out, it was a while ago now, maybe it's 8 years or 10 years ago, where we show that staph eats our blood to cause disease. And the amount of press that generated around potentially like explaining why bloodletting works and how bloodletting might be a strategy to protect against infection. It was a really interesting discussion that came out of that. But it's the balance that's needed and the impact that excess or deprivation of iron can have on the host microbe interaction is really profound. So I think you're right to think about it like that.
What about, so just yesterday I did a fascinating show about why sleep deprivation kills us. And the reality is in the context of the modern world where people either have lousy sleep or sleep very little to begin with, and how that actually accelerates disease states. And the interesting thing that came from this research was they were able to pinpoint the death of the fruit flies and the rodents by when their guts started to accumulate massive amounts of reactive oxygen species. Now, ROS is a very important molecule. We don't want to eradicate it. It kills the beginning of tumor cells. I mean, it's a good thing. And it's a signaling molecule, but it's also a mediator of inflammation. People don't think of oxidative stress as being a co-partner in the inflammatory process, but it really is. When we talk about oxidation, we're talking about inflammation kind of as an adjunct. But they found out that sleep deprivation causes a massive amount of ROS accumulation in the gut specifically, not in any other tissue. But then that triggers all this tissue damage throughout the body. Could it be that the people who have the 20% reoccurrence of C. diff, because we are talking about an elderly population, they suffer from things like sundowner, they don't like to sleep, they don't get good sleep, they're night owls. Could the lack of good sleep hygiene be predisposing them to a reoccurrence?
Well, I think it's worth thinking about and worth talking about. And it really takes us back to how does this disease happen? And the most important critical piece for a C. diff infection is disruption of your gut microbiome. So anything that disrupts the gut microbiome is going to potentially cause the ability of C. diff to colonize and cause disease. And we've all had these nights, right, where we stay up too late or we don't sleep at all or we pull an all-nighter. And one of the most common things that happens to me is the next day my stomach hurts, right? I'm screwed up. And so it's a bit of a stretch to say that people that don't sleep are going to be more susceptible to C. diff, but I don't think it's a bit of a stretch to say that people that don't get sleep have disruptions in their microbiome. And I know that people that have a certain disruptions in the microbiome are more susceptible to C. diff. So I definitely think it's worth talking about, and it's probably worth investigating in more detail. Absolutely. And this piece with the reactive oxygen as a component of that, I think it ties into this finding we talked about today is when you have inflammation, the first cells that come in are macrophages and neutrophils. And one of the biggest weapons that they have, which is what you mentioned, is reactive oxygen species. They mobilize reactive oxygen species to kill whatever they're trying to kill, be it a tumor cell or a bacterium. So you're going to have huge amounts of ROS in your gut if you have inflammation in your gut, regardless of how it was caused. And I think sleep deprivation could possibly be part of this.
So the video platform for some reason has some wonky security setting that certain people who watch the show live, they just show up as Facebook users. So anybody who's still watching, if you can post your name so I can give you credit for this. He says his wife has super high iron, and then he goes on to say from transfusions. Love your show. So is there any linkage between different medical therapies and the occurrence? I get the antibiotic thing. Are there any other correlates for people who have a higher risk of C. diff or higher risk of reinfecting?
Yeah, there are some sort of small studies that I think need to be repeated at larger levels that do show that transfusions can increase the incidence of C. diff or the severity of C. diff infection. So it's not unprecedented to talk about if you're, especially if the transfusions are changing the metal levels in the host, the person in this case. I think that can have a profound impact on the outcome of an infection.
And then Elisa Profumo, who you know because she established this interview, she's my executive producer. She asked probiotics. I would imagine probiotics would be good if we knew which ones we needed to replace, right?
Yeah, the two things that we need to know, the first is which ones do we put in, and the second is how do we get them to stay.
Yeah.
And that those are really challenging problems. And I think we as a community of researchers and people trying to solve these problems are honing in on the critical microbes and honing in on some strategies to get them to stay in our gut. But right now it's very hard to put one or two individual species into your gut, like orally, and have them actually take hold. That's a very hard thing to accomplish.
And this is an interesting, because it's worth talking about a little bit, right? So we a lot of us take probiotics, and most of us get in trouble with them. I did a show a few years ago that showed that the propensity to supplement with lactic acid producing bacteria, which is the most popular probiotics, actually could be leading to small intestinal bacterial overgrowth. And the show that we did showed that treating with an antibiotic that's used for traveler's distress. You go to Mexico, you eat the food, you get diarrhea, you take this for 3 days and it goes away. But for longer terms, I'm trying to think of the name of it. It starts with an X, Xifaxan. By taking Xifaxan for like 40 days at 1,500 milligrams a day, completely reversed the small bacterial overgrowth. And we knew this because this bacteria produces a type of lactic acid called D-lactic acid, which is different than the lactic acid that our muscles produce. So they were able to identify it and then track it. And it was successful because the D-lactic acidosis dropped. D-lactic acidosis was leading to brain fog. Everybody complained. So my point of this whole dissertation is, we take probiotics today like we think they are doing something specific, but we really don't know what they're doing. And more importantly, as you just pointed out, a lot of the research no one talks about, you put them in here and they come out the other end. They're not taking seed in your gut at all.
Right. Yeah. I think you've hit on some really important points. We're talking about an entire organism, right? A probiotic, pick your species, whichever one is marketed at whatever level. This is an organism with 4,000 genes that makes 4,000 different proteins that probably has the potential to affect your body in a number of different ways. And in very few cases do we really have any idea what those ways are. And not only might they affect your body directly, they can affect the other bacteria that are in your gut, which you may want there, or maybe you don't. There's very little information on exactly what all of these probiotics do or could potentially do, because we just don't know that much about the individual organisms we're talking about. And secondly, we just can't keep them in. They come right out the back, just like you said. So this is the huge challenge to the probiotic industry.
One last thing before we take our first break, and then we come back and I have some interesting topics to discuss with you. Because this is a spore, it won't be beneficially affected by diet. We know that extreme restrictive diets like the keto diet, maybe even the vegan diet, or now the carnivore diet, have the ability to change the diversity of the gut microbes and even put autoimmune disorders to sleep. But when they're spores, they can actually go into a state of suspended animation. So they never die. They just wait to be fed again so they can wake up. Is that accurate?
Yeah, so the spores are extraordinarily hard to kill. So with C. diff is in its spore state, there's very little, I think, that you could expect could happen to the organism that could kill it. The trick is once it vegetates, then it sort of shows its weaknesses, and then we can kill it either with antibiotics or immune cells. So one strategy would be to prevent sporulation. So if you could keep the organism from sporulating and keep it in a vegetative state, then that makes it easier to kill. But if you have spores, in a hospital, this is why it's such a tremendous hospital pathogen. Is the spores could potentially be all over the hospital, and then a person ingests the spores, and then they get the disease. And it also makes it harder to get rid of them, like you mentioned, which is one of the reasons why people have these recurrent infections.
Well, in fact, the interesting thing about the study we did on small intestinal bacterial overgrowth and Xifaxan was that the good doctor and I communicated after the show, because I told him I was going to run some Xifaxan to see if it helped, and it did help, in fact. It changed my gut completely. This distension went dramatically away. But he told me, don't stop eating what you're eating. He said, because we don't want any of the microbes that are spores to go into suspended animation. If you change your diet and you start to starve them, they'll just go to sleep to conserve energy. We want them to eat the antibiotic along with the other stuff they eat. So he insisted that you keep eating what you've been eating while you apply the therapy. So these spores don't go hide. They stay out and they can be killed.
Yeah,
I thought that was very interesting.
It is interesting, and it's smart, and it's a good way of thinking about things. And so there's other organisms that don't sporulate but go into a suspended animation, like tuberculosis does this. And there's a lot of investigators that are trying to keep TB from doing that. So when you add antibiotics, they actually work, because once these organisms when they're not replicating, they're not going to be susceptible to antimicrobials. Antimicrobials are all based on the organism replicating.
We're talking today with Dr. Eric Skaar. He is from Vanderbilt University Medical, and we're talking about his recent study on a possible way, now that we understand that C. diff uses heme to cloak itself against the immune system and against therapies. There might be ways to take advantage of that. I want to talk about some of the ways to take advantage of it that you spoke about in the research. Stay tuned. We'll be right back with more Superhuman Radio. We just have to we're going to roll on, and I'm going to cut the spots in later. We have a computer problem right now. That's okay. So we took a break and we're back. So talk about let me go back to to my notes for a second. So talk about the ability to use a protein. I think it was a protein that you said that would actually keep the C. diff available or out in the open, so to speak. This is something that could become a future therapy, right?
Well, we think so. So this protein HSMA, the way it works is C. diff senses blood, senses the heme in blood, turns on this protein, coats its outside of the bacterium in this protein, and the protein binds the heme from the blood. So it steals heme from the blood, puts it in the protein, puts the protein on the outside. Now, heme is really reactive, like we talked about earlier. So this is a positive aspect of heme. It's why it's such a valuable molecule for our bodies, but it also can be used against us. And in this case, the reactivity of heme is used to detoxify redox active molecules that normally would kill the bacteria. So when redox active molecules from either our neutrophils or our macrophages or antibiotics, like we talked about, when these things come after the bacteria uses HSMA to just detoxify them. So it's creating this sort of cloud around it of protection, so that none of our typically very powerful antimicrobial strategies, either immune or antibiotic, can kill it. And that, I think, is druggable. So if we could prevent that from happening, we would make the organism more sensitive to our immune cells. We'd make the organism more sensitive to antibiotics. And there's two ways that I can think of to do that. The first is you prevent the protein from being made or going where it goes. So we could develop an antibiotic that targets HSMA. The second is without heme, HSMA is useless. So if we could keep heme away from C. diff, I think we also would have a protective strategy.
Now, the reason this is actually viable is because, as you point out, the way it gets into us through the gut. So you're not going to alter heme's activity in all tissue throughout the body. You just have to alter it in the gut, right?
That's exactly right. Yeah. So you can imagine like an orally available heme binding protein or heme sequestering complex or something like this alongside of an antibiotic could potentially make that antibiotic a lot more effective against C. diff.
Is there anything that binds heme now?
Yeah, so our body makes a protein that does exactly this for exactly this reason. So one of the most powerful arms of the immune system that doesn't actually get a lot of attention is something called nutritional immunity. And so this is the idea that our body makes proteins to keep food away from bacteria. And you probably heard about them, you just may not have thought of them in this context. The most well-known one is transferrin. So this is a protein that binds iron. And so when we get infected, our body makes more transferrin. Transferrin binds more iron to keep it away from bacteria. Heme is the same thing. So heme is such a rich iron source for bacteria. Our body needs a way to keep it away from bacteria. So we make a protein called hemopexin. Hemopexin is a very high affinity heme chelating or binding protein. And you can imagine that potentially a molecule like that, which we already put into put into animals for various reasons, could be used to sequester heme and make C. diff more sensitive to antibiotics.
So something that's available for nutritionally applied processes is found in bovine dairy, and it can be isolated through molecular sieving, and that is lactoferrin.
Yeah.
And lactoferrin will bind to iron in the gut and literally carry it out of the body. It'll keep you from absorbing. Other things that will keep you from absorbing iron are epicatechins in green tea. Some studies show even coffee, but I'm not a big proponent of coffee because I think it actually is part of the problem we have today in our society is the high amounts of coffee consumption. So what about lactoferrin and things like epicatechins? Could they be possible therapeutic aids?
Well, yeah, lactoferrin is one of the most, along with transferrin, it's one of the most important arms of the nutritional immune response. So absolutely, this is a molecule to think about. The thing that makes it a little tricky in this case is we're not just talking about iron, we're talking about heme. And lactoferrin will not bind heme iron. It only binds free iron. And transferrin only binds free iron. And the iron in heme is bound so tightly that these proteins can't pull it out. So that makes it a little bit different. We need something that actually binds the heme, because without heme, C. diff can't make heme. So even if you give it iron, HSMA is useless. The bacteria has to see the heme molecule.
So what would you like both clinicians and lay people to take away from this current research?
Well, I think that one thing that's potentially really interesting, and this is speculative, but it's interesting to think about, is it's never been abundantly clear why C. diff makes these toxins. There's organisms like cholera that make toxins in our gut, and it makes a lot of sense because cholera causes this massive watery diarrhea, so presumably the bacteria can get back into the water system and infect more people. C. diff, it doesn't make a lot of sense why C. diff wants all this damage and inflammation. And one interesting possibility might be it wants our blood. It wants the heme so that it can make this shield. So now it's protected against all these antimicrobial molecules. And again, that's speculative, but I think it's a really interesting possibility and something that we're exploring in our lab. The other thing I think that is worth experimenting, and again, I'm not suggesting I know this is a good idea. I just like the concept, and I think we should investigate it, is how much heme is available in the gut during a C. diff infection? Can we reduce it? And, could that be, should we maybe think about less heme in our diet for a person that is suffering from C. diff or at risk of C. diff? Should we come up with sequestration strategies like we just talked about to keep heme out of the diet? Is there a way to prevent blood flow into the gut somehow without disrupting the immune system? I mean, these are challenging questions, but I think the end result is if you can have less heme in the gut, when you hit with antibiotics, you're going to have better treatment outcomes. So that's what I want to take from this.
What about a suicide molecule? What about something that looks like heme and the C. diff thinks it's heme and it binds to it, but it's not heme and it just kills it, or doesn't kill it, but it just keeps it from attaching to heme and it dies, or it's more vulnerable to antibiotics.
Yeah, we should do this. You should come in the lab. We could do this. So there's heme. The chemical name for heme is protoporphyrin 9, and if it has an iron in it, it's called iron protoporphyrin 9, and that's what heme is. And you could swap that iron out for other metals, and then you get gallium protoporphyrin 9, tin protoporphyrin 9, zinc protoporphyrin 9. Those molecules will not do what heme does. So those would be exactly the kind of molecules you're talking about, a decoy molecule. So presumably, if you fed C. diff gallium protoporphyrin 9, it would eat it, would think it was heme, it would eat it, it would put it in HSMA, it would put HSMA on the outside of the cell, but it wouldn't be protective. So that's definitely a cool idea.
That would be great. I hope you do that so you can come back on the show and talk about it. That would be great.
I'll give you credit.
Listen, we have a little technical difficulty. We would have run 2 commercials during our break, and I'm going to have it inserted in editing later. But I think your work is fascinating, and I think it's important because we just went through this big process here in the United States, not only to control the widespread of a virus that would have overwhelmed our medical system, but also because we know that this particular virus, COVID-19, preys on the elderly. And I think it would be a fair thing to say that C. diff preys on the elderly. It's not a disease of young people. It's a disease. It's a problem with old people. And it's generally a problem with old people who have the least available resources. They're usually in institutions or hospitalized for long periods of time. So this means that they are very ill. They're very weak. And this opportunistic bacteria literally just pries any life that they have left out of them. And it's a horrible way to die. My mother was dying from it until we had this doctor who said, there's this new thing. Let's try it. Fecal transplant. Do you have any relatives that live here? Yeah, my niece donated, and boom, my mother was alive.
Yeah.
I think your work is great. This is not a sexy discussion. No one wants to talk about C. diff, but the reality is it kills a lot of elderly people and it shortens their lives unnecessarily. And we should take the same approach and interest in it as we just did COVID-19, I think.
Yeah, I mean, it's like you said, it's a huge public health problem. This is it's not something that only a few people get either. Granted, the elderly are at increased risk, but we're talking about a lot of people. Number 1 cause of hospital-acquired infections in the U.S. So this is definitely, I agree, it needs more attention.
Thank you so much for being on the show.
Thank you for having me. I really appreciate the opportunity.
Take care.
Bye.
All right, so normally I would take a break here, but I'm not taking a break because I can't run the studio server and put my commercial breaks in. If you're listening to the podcast, you're listening to commercial breaks because I found a way to insert them. But I want to go ahead and go on to the next area of discussion. I want to take this down first. I'm doing stuff that the podcast audience can't appreciate because it's all visual. And I want to talk about some critical thinking. I like this, Critical Thinking 101. Little thoughts that I have that I think are valuable. And, maybe you think that this is minutia. Maybe you think this is nonsense. But if I can provoke you to think differently, then I've done my job. So I was looking at the etymological origins of the word allergy. Okay. The word allergy comes from the Greek word allos for other energy. Then the German language mutated it into ALERGIE, but it still pretty much meant the same thing, other energy. And then eventually it became to the word that we have today, ALLERGY. If you look at the original origins of the word, what you discover is early scientists and science-minded people believed that the body at homeostasis wants to remain at homeostasis. Any interruption of homeostatic tendencies was thought of as an other energy, taking you in your body in a different direction than where it wanted to be. So somewhere along the line, we decided as humans what were good allergies and what were bad allergies. So, for instance, if you happen to catch poison ivy, that was a bad allergy, right? Because you broke out in hives and you were irritated and you didn't feel good, and this was a bad allergy. But somewhere along the line, we decided other allergic reactions would actually be beneficial to us. Caffeine, for instance. Caffeine works because you're allergic to it. Think about that for a second. I have friends, Ron Penna, one of them. Ron Penna can take caffeine. He has no effect from it. Doesn't keep him from sleeping, doesn't give him energy, doesn't give him focus, doesn't give him the jitters, doesn't affect him. So genetically, maybe he's someone who clears it quickly. I would submit that his body works harder to remain in homeostasis. I can't stand caffeine. I've been addicted to it numerous times throughout my life, and it always ends the same way. Makes me feel horrible. But for some reason, we decide that what caffeine does is more desirable than, let's say, what poison ivy does. Right? Caffeine elevates your heart rate, increases metabolic rate, gives you anxious, nervous energy, improves focus. And from a standpoint of today, modern society, these are good. Oh yeah, we want to be up. We want to. But let's look at it from an evolutionary perspective. What would a human a million years ago done if they ate a plant that made them feel that way? You think they'd go back and eat it the next day? Maybe. I don't think so. I think that they would have been so freaked out by how they felt and anxious and nervous energy that they would have thought, oh, that's poison. Don't eat that. Don't eat that. But then because of customs and modernity, we decide that that's what we need. And so now we look at caffeine and its ability to cause an allergic reaction as a good thing. It's desirable. So I want you to think about this in the context of your own life. There are things that you do day in and day out that are actually working the way you want them to because you're allergic to them. Caffeine is just a low-hanging fruit, easy for me to point at. A lot of people use it. But there are others. And some of them, we've now called them biohacks. You know, those of us who take nootropics. I would submit that the reason they work is because they're causing an allergic reaction. They're causing a reaction that goes against homeostasis. Your brain doesn't want to be that way. It wants to be this way, but you don't like the way your brain is now. So you want to change it. So you use something that actually causes an allergic reaction in the true meaning of the word, other energy. And maybe we need to come up with a new term for allergic reaction. Maybe we need to come up with a new term for allergies. Maybe we should look at allergies as the ones that are desirable and less desirable. And I submit the word epithergic because epithemidos is the word for desirable in Greek. And since these words all started in the Greek lexicon, epithemidos is the word for desirable. So desirable energy. We desire this change in energy. We want it to do this for us. So we embrace it. So I think epithergic should be a word that should be added to the lexicon when talking about science and medicine. That's it for my Critical Thinking 101. There are a lot of things in your life that you like, the way they make you feel, but that's because you are allergic to them. Think about that. And that's it for today. Sorry for the technical difficulties. Yeah, you ended up with a shorter show and all the information you need. Thank you for listening today. Thank you for watching. And we'll see you tomorrow with more Superhuman Radio. And I can't even run the end music here. How do you like that? I'll sing. All right.